Very good evening, everyone. I hope you are all doing great. So we are done with our basics of molecular biology. So we're gonna start with some techniques today. That is your microscopy. So in the basics of microscopy will be taken care. That is that how this is from the plus two. Before I go to the advanced microscopy, we will be discussing about confocal microscopy, electron microscopy, light microscopy, many more. But I just want to show you what is the basic microscope looks like that we tend to use in our 12th class biology laboratory practicals. We tend to go there and we used to see some like slides, fixed slides from pteridophytes or some mitosis, the fixed slides we can see at that time. So microscopes. So basically microscopes are used to magnify objects which we cannot see with our naked eye, and which help scientists to think how actually they are working in real life and what is the real shape is in the real life sense, in the live imaging. So basically on the top of the most of the microscope is the ocular lens, that is your eyepiece, then body tube, and there is a nose piece, objective lens, then the stage clips are there with a diaphragm and the light source. Then there is a base also at the bottom with the fine adjustment, and coarse adjustment is there to make finer adjustment. Then we can use this analog. Then for better broader adjustment we use this one, and stage and then on. So that's the basic structure of our lab day-to-day life microscope. So there are three types of microscope. That is simple, which has only one lens, right? And then there is a compound microscope which has two sets of lenses which can magnify from 100 times to 200 times. And then there's electron microscope which can magnify our objects up to 300,000 times. So they do not use lenses, but they use electrons to enlarge the image, to magnify the image, right? So parts of microscope. So first is your ocular lens, that is your eyepiece, through which we see through the microscope. And then coarse adjustments are the one through which we, with the knob, we focus our samples. And then fine adjustments is to bring image into the focus. So these are placed according to this. Then there's an arm to hold the body tube. Then there's a body tube in order to join your eyepiece and objective lens. Then there's a nose piece at the bottom of your body tube of your microscope, which holds the objective lens and allows them to be turned. Arm, body tube and nose piece. Then high power objective lenses are there. So this could be 40x, then could be low power objective lens of 3x and x, 4x, or could be middle power from 10x to 20x. So you can remove them in the circular order and the clockwise or anti-clockwise order. And then first you start with always with the lower magnification. You fix what you want to look for, then you go for higher magnification. Then the last stage is your stage on which you put your sample. Then there's a clip to hold your slides, and there's a diaphragm for the light entering into the field view. From the bottom, from the diaphragm, your light source is bringing to the up. The light source is there, which is like could be lamp or mirror. So if it is a mirror, we can use the sunlight light or normal tube light in order to make our image like brighter. And there's a base at the bottom to hold your whole microscope. So that's the basic anatomy of our microscopes looks like. And based on this microscope, actually your all advanced microscopes are made up of. So this is trivia now. So what we have discussed, we will go through them one by one. So first is your ocular lens, then this body tube, nose piece, objective, stage clips, then this is what is it? It's diaphragm, then at the bottom is your light source, then the arm is there, then the stage, then coarse adjustment, fine adjustment, then base. And from here we tend to hold our microscope. This part we put our hand in it and then we go for it. So how does the image looks in these microscopes actually? So field of area in the circle, that what we see through the eyepiece. If E is written like this, so we will see of the same thing upside down and the mirror image. Upside down and the mirror image. So that's what we see during the field view. And we can also compare the magnification, like if it is 7x, this eagle's eye, by the 10x it will be more magnified. Same, there is a lymphoma of some blood cells here, and we can magnify by 10x even more better. So that's the significance of our microscope is. So first you focus something that you are looking for, then afterwards you go in deeper part of it and then check the morphology, how the cells looks like. So the normal cells, if you see, they are very fine. There is nothing coarse like these. These cells are very broader. They are big, and they are more pinkish as compared to the other one. So this is the advantage of the magnification within the microscope. So what is my power? So power could be, which actually means the power of magnification in order to calculate is to multiply the power of ocular lens by the power of objective. So 10x, 40x. So let's calculate the magnifications. So find the power of lens, it is found on the side of the lens. So on top it's written whether it's a 10x, 1000x, 40x. Then you multiply that power with the eyepiece by the power of the objective lens. For example, if the eyepiece is 10x and your objective lens is 100x, then you will have the answer as 100,000x will be your magnification. Yeah, this is your eyepiece, this is your objective lens, and you multiply these together, then that will be your magnification power. So as I said, objectives, the specimens that you are looking for, they are mostly upside down present and backward. So if E is like this, it will be upside down and it will be always be like that. So this, if we have a practical, then we can do this, but we don't have at the moment. So just forget that. So how to make your slide actually? So what we do is you take first your oil drop on it, then you put your sample on that, and then you put your slide with the 45 degrees Celsius, and then slowly with the help of tweezers you slide it down so that no air bubble should come inside. Gently put it on a water or its specimen so that things are finally be done. So cover slip, slowly you lower down it and then it's good to go. So that's it. Just want to give you a hint of microscope now. Now we will go to the advanced part of microscopy. So this is one funny cartoons, that is the scientist is looking in the microscope, 3 pairs of legs. So it's calling it as, oh, I found a mutant genes. So it's a gene which is having 3 pairs, and he's saying that, oh, it's a mutant genes. It's a nerd joke. Okay. If sir, if you say. Oh, good, good, good. So where am I? Yeah, I have to go to the files. So I will be now from now onwards a bit slow. I will speak slow, and we take things one by one microscope, one by each. Yeah. So light and electron microscopy. So basically, um, your microscopy, it's made up of it's of two types, has been divided. That is your light and electron. In previous time, it was very hard for the people from the 18th century to look the reason for diseases, the reason why somebody get that disease, and it was very hard to find a diagnosis. Why children's getting jaundice? Why they getting smallpox? So why there's a pandemic is happening? Because nobody can see these microorganisms. Nobody could see these viruses. Nobody know their structure. So nobody could work upon them. It's just the recent research from the, like, let's say 1950s, yeah, from the 1950s, where molecular biology, biotechnology started to develop, and with the help of bioinformatics also, uh, your microscopy techniques also develop quite advanced. So with the help of, with all these techniques, we are able to understand, so what is the reason for one particular reason, like for, let's say, GMO crops that we are producing. So we were not aware of these genes and the chromosomes, how many are there present, how can we increase the yield of one crop within the same field of hectares. So this was all things were possible because of the microscopy techniques that had been identified, molecular biology, your biochemistry, and the various techniques that has been discovered. So here we can see a replica of a Leeuwenhoek microscope from the 1670 that he has constructed, one example. So this is a replica of Culpeper tripod microscope, which was built in 1725 by Edmund Culpeper, and the collection of Moody Medical. It's there in the Texas, in the University of Texas there. So in the library, they're all present. So you can see it has objective lens. Yeah, here eyepiece, source of light, and you could put your sample here. So basically, it's also a microscope, but very fancy, wooden, uh, but working pretty good at that time too. And this is from the Marshall microscope by the John Marshall. A better having a base here, tarnished with the golden platelets here, in order to see the things better. So this is the contents of the microscopy that we will be discussing. We will be discussing about various types of microscopy, including your light and electron. So within the light, there are 7 we might be discussing: bright field, dark field, phase contrast, polarizing, DIC, epifluorescence, confocal laser scanning. Then within the electron microscopy, we might be discussing about scanning, electron microscope, SEM, and transmission electron microscope. Both have different principles. And then later on, we will discuss about how we prepare specimens for these different type of microscopy, for electron microscopy and for your light microscopy, how the specimen different preparation is done. For the specimen preparation, I had a quite 1 year of experience in the histology lab, and this work, to do this work, you need to be very concentrated, focused, and it's a boring work because you have to just repeat. You just have to repeat and repeat and repeat. That's it. So most of the time, science is pretty boring. You do the experiments. You have to do these experiments at least 4 to 5 times in order to get the right results. Yeah. But once you get the results, once it started to make sense, once it's making a story, once everything is coming into the puzzle, things starts to fit. then it's a different level of excitement, different level of boom in the science. Yeah, so we will be discussing these things. So let's start with the an usual microscope. This is an old molecular bright field microscope. So in the old time, very old time, that's how they look. Maybe from the World War time, I think this is used to be. So microscopes have long been essential tools of cell biologists, and in this tutorial provides a brief overview of the microscopes commonly used in the biological studies and journal techniques for preparing specimens of various types of microscopy. So the two broad, as I discussed, we will be discussing here the of microscopy is light and electron microscopy, that is in the market now. So let's start with the light microscopy, that is bright field microscopy, that is mostly that we are using in the lab. Once you enter into the lab, into the molecular lab or CSIR lab in India, or any big institute, ISR labs, ISC lab, IIT's labs, anywhere in the big world or in some private institute, this is the microscope that you will see most commonly being used by the scientists, students, anyone. Most of the work, 80% of work is done by this simple microscope. Yeah. So this is the most common generally used microscope, bright field microscope, that are named because of the field is bright light in this. So field light is bright light here. As the one that we were just discussing in our previous basics microscopy, while the objective view is dark in this. So simple in this is a binocular, so it has not single piece of view. So you can have a view with the both eyes. It's much better. Yeah, it has movable stage. So this stage, when you put your slide here, so you can move this slide horizontal and vertical very easily. Yeah, and it has iris diaphragm, then condenser, and there's a built-in light source. So you don't need there is no mirror is there, so there is a bulb is being there fixed down, so it's giving a light source of it. So these types of microscope, as I said, used in the clinical research and students microscope. So they have very simple design. Light directed at specimen is absorbed to form image. Unstained. Yeah, they are unstained. Specimens have poor contrast, and stained specimens show excellent contrast in them. They are ideal for stained bacteria, cells and tissues. They have very high numerical aperture and good resolution, and quite bright background, and for dark specimens. And the light source is tungsten and halogen in them. So that are the pretty much characteristics of this light microscope. Now we have discussed about this microscope. Now it's time to see how this microscope looks inside the samples. How does the samples looks of them? So these are the images. As I suggested, it's a bright field, so you will see light in the sample, of course. You will not see dark background anymore. You will see big thick light at the background, which is somehow not so good for focusing your things that you're looking for. But on the other hand, in order to identify things that you're looking at the primary level, it's okay. You can go ahead. But if you want to publish this data and want to go for higher general, most likely you have to go with the confocal microscope or higher end microscopy. But these are acceptable at the beginners level. So there are many stages when you want to publish, when you want to be author, like a good scientist. So there's a thing called a good scientist, a good researcher, and a bad scientist and a bad researcher. Yeah, for your whole life you can be professor. Anyone could be professor. Yeah, there is no problem. But the problem is, how good are you ethically while working with your work? How you do your work with full of truthness, and there is no hidden gimmicks in it. So that matters a lot while you're working, because there are a lot of retractions are happening because people put their results in the paper, and somebody wants to repeat those experiments. If somebody says, so let's say, I don't know what these figures are, but this is the ciliated parasites which are containing the gut tissue, but there is nothing like disease state or unaltered state. But if somebody want to say that the heart sample looks, the morphology of the tissues in the heart failure samples, it's look like that, and in the case of giving the drug, they have recovered that thing, and somebody could alter that thing for the just publishing thing, and it could not be repeated by another scientist, so that paper will be retracted means it will be not published, and it's a lot of embarrassment in the scientific world. So whenever you want to start your work within the science field, I will highly recommend do your things with full of truthfulness. What you see, say that. Don't say that to make your boss happy or to make audience happy, to make scientific world happy, to make your research an extraordinary. Because if it is not true, it will eventually people will get to know later. Not now, not in 5 years, but in 10 years for sure somewhere. And once it is fine, whole career is duped out. So I'm saying this thing, I'm sharing this statement with my most of the students, from my own experience. um, when I was working in the Max Planck Institute of Heart and Lung Research, so I was told by my boss actually, so if you want to work ethically on the long term, you want to be success, a short term success is very easy, you can alter the results, but on the long term, if you want to live a longer and healthy life and with lot of your own guts, that we should do actually. So by the end of life, we should have no regrets. Yeah, so that's the bache I'm telling. At the moment, you all are studying at undergraduate or graduate level, but I'm saying these other stuff that by the end of your life, well, the work you will be doing in the future, in whichever the lab you will be working, it should be ethically correct, so that you know that what you did is right and there is nothing wrong in that. So you will not have a regret at the end of the life. So this is the stained blood cells in peripheral blood smear. In these cells looking like this, and this is flagellate trichomonas, with having this peripheral outside, flagellum and nucleus inside, and so on. And this bright field microscopy could be also be modified, and that could be called as inverted microscope, that we could see in the right figure. Here the light source is from the top, yeah, and the objective is inverted. So objective lenses are at the bottom, and the eyepiece is towards your eyes, binoculars. So that's something inverted microscope. So this allow viewing of cells in flask, well plates, or other deep containers that do not fit between the objective stage of standard bright field microscope. So sometimes you have to work with 6 well plate work, 6 well or 96 well plate or 8 well plate cell culture, and you want to analyze each data and capture those images. So there are chances, there's a computer is attached to them. It's by the Alicia. We used to have Alicia inverted microscope. If I could show you. So they have actually, no, not this one. Okay. Yeah, what we do in that inverted microscope, there is a closed chamber, yeah, and we put a carbon dioxide source, oxygen source, and we don't let yeah, pressure is maintained, and everything is maintained so that when we put our cells there, they get the very same culture conditions that they were getting in the incubator. uh, your CO2, temperature, pressure, everything is maintained same way. And you can run your samples for 24 hours for live cell imaging. You can run for 24 hours for live cell imaging, which is very, I mean, I want to show you actually, if I could show that how these platforms looks like. Inverted microscope with the Licia, not even Licia, if something else is there. So it will be very good to show you if you got these um, select all.
Advanced wide-field live cell imaging systems often have limited flexibility, allowing only one type of fluorescent imaging technique at a time. The DMI-8S microscope from Leica Microsystems eliminates this limitation. With the exclusive Infinity Port open platform architecture, you can easily combine applications by adding one or more advanced fluorescence imaging modules, such as the Infinity Scanner and Infinity Turf. These modules allow you to expose your sample to multiple techniques simultaneously, giving you deeper insights into your cells. Add the Infinity Scanner to the Infinity port of the DMI-8S microscope and enable a host of photomanipulation techniques. Perform FRAP, flip, photo activation as well as photo switching, optogenetics, and ablation. You can also advance your research by combining several photomanipulation modes in one experiment. For example, apply photo damage to the nucleus to induce DNA damage. Then use photo switching to identify and track when repair proteins are released from the DNA. In addition, the Infinity Scanner is seamlessly integrated into the Leica LASX software. Setting up and running experiments is straightforward, allowing you to see the hidden to advance your discoveries. The Leica DMI-8S offers near infinite possibilities to adapt and expand with your research and enable your discoveries today and tomorrow.
So it was a very advanced microscopy that they were telling. I was not aware of that. Um, this is a recently they could, as they suggested, ablation, photo activation. All these things could be done with the help of that. This recent production of this microscopy. So yeah, it has indeed a lot of advantages to the another one. Now comes the next part, that is, yeah, I want to show you how the images looks like in them, in the dark field, or sorry, in the bright field microscopy. If you run them over the live cell imaging, how the results looks like. Yeah, so I did experiment in 4 different wells in a 6 well plate. So I got my neonatal red cardiomyocytes and I put them, yeah, and grow them over the cell culture plates. And then there is a conditions that I gave it. So I put the first condition was control, and then I added 100 micromolar of peroxide, hydrogen peroxide. Then there is a 1000 micromolar of peroxide, and then there is a 10,000 micromolar of peroxide. Right? 100, 1000, and 10,000. Yeah. And then what you do is you do the physiological effect of redox regulation in the cardiac myocytes. Then I want to show you these live cell imaging which was done for 24 hours, and then this is the things that we see. So in the control, cells were fine in 100 micromolar of H2O2. So I want to check the effect of redox on the cells. What happened if we give hydrogen peroxide an oxidant? So nothing happened at 100 micromolar, but at 1000 cell seems to act different. At 10,000 micromolar, all the nucleuses start to pop out from the nucleus. So these are the live cells, neonatal rat cardiomyocytes, showing sorry, different characteristics, morphology at the different concentration of hydrogen peroxide. So you can do the same experiments, with any cell line, whether it's liver cancer cell line or skin cells, liver cells, lung cells, kidney, any cells that you can, HEC cells. So all these things are possible. So that's what the images look like. And on the right hand side, it's a MATLAB software. This is also very in if somebody is into the research already. So this is also in nowadays. So nobody accept these results. Nobody want to see this actually. Nobody cares about them, In the research, nobody cares what you see. You have to represent that figure into the graph. What actually you see in the quantitative or qualitative way. Yeah, what do you see in the quantitative? What do you see the differences? So in this MATLAB software, it's calculate the contraction of your heart cells. That how many of your cells are contracting. So what we see that in the mean contraction, there was no changes, as I said, in these two, in the first three. In the fourth one, it was drastic change. There was no contraction was happening. So this was done by Hubish et al. He helped me in this project, and then we were able to make this graph. And students, you might be having these courses called biostats. Yeah. So I just want to, because this topic has been started, it's always good to share with you, not to be very specific with the topic that we are discussing, but also something allied things. So in your biostats in the college, we usually are teaching students like ANOVA test, F test, T test. There are many tests with which you can check your statistical analysis within group or so. And we might be teaching you, I was teaching actually biostats in my college. So what the thing was that we were teaching them with a very traditional and very boring way, to be honest. We were not allowed to use softwares, the computers. So we have to give them these tables, these figures, and you have to do it manually. And it was very complicated. I don't know why we have these all systems still in this curriculum. So there's a software called, I don't know if still I have, GraphPad Prism. I don't have this software now. So you can download this software Prism, and if it's possible from the university, you can do it. So this software will do your analysis automatically for you. You don't have to do those complicated calculations on your lab book. So in my PhD, I never used my lab book, to be honest, telling you. Everything was done on the laptop itself. All analysis, all data, everything was saved over the computer. So this will make your graphs, very nice and paper presentable. So all this is the blue lines is the with inhibitor, and this is the non inhibitor, and this is the y-axis, this is x-axis, and yeah, it's there. So in order to check the mouse samples, mouse treated with and control. So in this way we can group them in order to identify the peaks, how the things are going on. So this is it's a multi-purpose software and makes your life easy. And there's lots of softwares are available in the um, I will share this. There are lots of like online tutorials are there. So just sharing you so that your life could be easy. Don't go for the complex world with the some people do all these calculation over the excels. So this is the most traditional way they were doing, but that's, to me, I think it should not be the ideal way, because excel could do sometimes errors, but this craftpad prism never show any errors actually. Saurabh Singh, how can we distinguish between tissues just looking at the histology? So, bache, there's a lab called pathology lab, and these people, they are the famous, like they are the professional pathologists, and they had this training from many years to see cells, to see tissues of disease state or a non-disease state, many things. So just with their own experience, they can see it. But for the newcomer, I know it will be very difficult. That's why if somebody have taken a medicine has taken as an MBBS, so there are courses in the microbiology. They are shown these images. You have to remember these images, that how a particular squamous cell line or heart cell line or your nerve cell lines, how these all look. So you have to be practice this. This doesn't come in a single day. This takes some times. So this was my PhD work. Yeah, I want to show you these things. So this I did, these figures that you're seeing here are also the neonatal red cardiomyocytes. So I did a siRNA transfection, in order to decrease the knockdown the effect of one protein called CTGF. So in this we are seeing control, and this we are seeing CTGF. And I've added fluorescence dye, that is as a DAPI. DAPI is used to fluorescent your nucleus. And ATF6, it's a one protein, is used in the ER stress marker. It's present initially outside the Golgi body or nucleus. And CTGF also present outside, and this is the merge figure of DAPI, ATF6 and CTGF. But once the knockdown was done, so this ATF6 tends to go inside, and there's a knockdown effect of CTGF could be seen here, that there decrease in the expression, and we can see also the merge image that most of the things, they are in the center, not on the outside like in earlier case. So in this way, you can predict many things, you can answer things from the cell perspective, that what you are seeing is right, and there is nothing wrong in that. And this makes your data more stronger, more applicable for the science, and people tend to understand what you are saying. Yeah. So the techniques that we are going through during this workshop is mainly preparing to how to do it. So these techniques even a technician could do, believe me, from my own experience. So any technique a technician could do, and it's not a problem. But being a scientist, if you are going for PhD, being a research associate, You have to think smartly, you have to think differently, you have to be clever in order to do your experiments using these techniques within the specific time, and you want your results to be positive. Yeah, you don't want results that you're not getting the results after doing first experiment, and you need to be very sharp that at the first when you do your experiments you're getting your results and good to go for further. Okay, now let's take the part of dark field microscopy. So this dark field microscopy, in this the background is dark, which allows the viewing of a small unstained objects such as motile bacteria that will be difficult to view in the bright field, and the central portion of the light is blocked so that only oblique light strikes the specimen. So this is a method from the 19th century. Bright specimens, dark background, and light not scattered by the specimens bypassing the objective, therefore making the field dark. Can see very small objects, but resolution is variable. So high contrast, good for unstained, live and motile specimens. So here we can see in these specimens the background is pretty much dark here. This is the algae hydrocyton reticulatum viewed under dark field microscope, and on the right hand side is the spirochete viewed under the dark microscope. The background is dark, so your images are more brighter and you can analyze things in better way actually. So it has somehow advantages over the bright field microscopy. Then there is a phase contrast microscopy. In this phase contrast microscopy, it converts the differences in the refractive index in specimens to differences in the image brightness. So central portion of the light source is blocked, yeah, and creating a ring of light from the condenser that illuminates the specimens. And the light waves refracted by the specimens and are slowed by the phase retardation plates. Right? So this is a technology from the 1940. It has phase objectives and provides higher contrast, good resolution. This is good for bacteria, flagella, cilia, organelles such as mitochondria. And they're good for unstained or live mounts. And yeah, this is the annular ring and this is the phase objective on top. So this is the phase contrast, and the images here, they on the left hand side, so this is the unstained squamous epithelial cells, that is your skin cells epithelial observed with the phase contrast microscopy above, and on the right hand side is the DIC. We will discuss this microscopy later, maybe tomorrow. So we can see that it's very fine, we can see the nucleus, everything is better here, more defined, we can see the cell membranes quite better. But the organelles, the cell organelles inside them, this membrane one and non-membrane one, they are not very well defined. We cannot say what is or what. Even though it's quite magnified, things are pretty much good, but still it might be not as good as will be another microscopes. So yeah, so the thing is, it depends what is your use is, what kind of microscopy in your lab is. Whenever you're gonna join any lab, just check what kind of microscopies they have, and according to that you do your experiments and get your results out. So sometime I was doing one experiments for one whole year, and I was not aware that there is a very nice microscope was present in my neighbor lab, which could help me to get a better results. So some of my friends just told me, oh, we can, if we just talk to the PI in that lab, we can get the very good results out of it. So that's also one thing. Don't be an introvert. Yeah, in the lab world, in the science world, being introvert could be also very harmful. I was very introvert, shy. I do not talk with anyone openly. So it could be a good thing for some time, yes, but could be also we can miss many opportunities, many things that we wanted to have, and just being a shy person, we cannot talk with the people. But I mean, A.R. Rahman is introvert, yeah, he's a shy person, and he's a maestro of music. So this things also doesn't matter much. but if you're master of something, you could be, and that point your introvert and extrovert also not work. So it's up to you how you want to take it. So I'm just stating a statement contrary to myself also. Then there is a polarization microscope. In this, we have a polarizer on top and bottom of a specimen. Yeah, so they help to increase the double fraction, that is the velocity of light refracted by substance, which is not the same in all directions. And the specimen is placed between two polarizers crossed at 90 degree to one another. So they are 90 degree towards each other. And we get a very bright image, very good dark background, and used for substances with highly organized molecular structures such as crystals, minerals. And it could be quantitative also. Yeah. So this is the images of polarization microscope. It's mostly used for the minerals from the crystals. Like this is from the Martian meteoroid, which was fallen on the Earth. Like from the Mars meteoroid came, and then we check them under this microscopy. So this one is the bright field of from that, and this one is the bright field from the renal epithelial cells containing fat globules, and this one is the high magnification of the same cells with microscopy. We just magnify these 4 different compartments, and these are called as Maltese cross. Actually, Maltese cross. So students, we do this epifluorescence microscopy tomorrow. Yeah, just tell me, I might forget from where we have to start. So that's it for today. So we had a good talk, not only scientifically, also personally. Diptesh, that's I'm telling this in these classes many times, that never ever use your images from Google or any general straightforwardly. You have to buy copyrights from them, or you have to make by yourself. So cells, tissues under face. So, bache, it's mainly all cells are possible. It's not very specific to it, but it's good for bacteria, flagella, cilia, organelles, such as mitochondria. Okay, class, you might be having next class of vaccine technology. So let's meet tomorrow, and then we talk about further about microscopy, some ethical issues that you have to take care, being a good scientist, being a good researcher. Yeah, because there's always one voice within us which says that this is right, this is wrong. Yeah, there's always one voice within us which says that this is right and this is wrong, and you have to decide what is good for me. Yeah, on that note, thank you so much. Take care. Bye bye. Good night. Stay safe.